ideaal.
Dat kon eens keer doen.
Hij dat niet livestream op telefoon.
Ik heb twee voor. Ik ga dan anders doen.
Mijn boekje, dat je zo moet trappen of voor de website.
Deze lang ga ik hem maken. Is goed. Ah nee, inderdaad. Die gaat vervangen worden. En dan zou dat weggedaan als een nieuwe wedstrijd. Mag zij daar een beetje water nemen? Morgen wordt dat waarschijnlijk toch ververst. Ja, doe maar.
Er staat ook nog koffie als er iemand nog koffie moet hebben. Water. Hij is misschien een beetje lauw ondertussen.
Ik denk dat we daar aan moeten van blijven. Ik zie daar nogthans zo kikkertjes lachen aan mij, maar... dat ik daar kikkertjes zie lachen aan mij, maar...
Dat is een ander iets. Dat is van onze studenten.
Nee, ik ga er van bij. Ik zou er niet aan. Ja,
Goedenavond, hè?
Ik ben niet ziek geweest, maar als je een week kwijt zet, dan direct. Ja. Dat heeft 4, hè? En een week of 4 hebt, is dat dan. Ik zit met iemand van Brazilië die testen bij ons komt toen ik niet verleid komt te zijn. Zei die maar een maand, ik ben er weer weggelopen. Ik denk zo dat de vorige examen is gehad, dan weet ik wel. En dan ook school dat ik kan komen. Dus.
Wachten wij nog op iemand?
Ik denk dat.
Dat is een select publiek.
Wat is dat? Isabel, nu ze komen. Dan zal het wel zijn, misschien. En dan Isabel is.
En laten we nu hopen dat de opname lukt, hè. De vorige keer konden we er echt niet aan doen, van we hadden de camera uitgeleend voor de proclamatie. En ze hebben bij de proclamatie de HDMI-kaart verwijderd, ze hebben de SD-kaart verwijderd, de HDMI-kabel verwijderd. En zo werd het gegeven bezonden materiaal, dus vandaar dat natuurlijk niet. Ik kon niet opnemen en het was dan ook niet verbonden.
Maar ik vind eigenlijk die screencast soms wel wat handiger.
Ja.
Omdat je meer detail hebt. Want bij sommigen, like met die kleuren, is dat niet altijd goed zichtbaar. Als je het film bent met de screencast, is soms veel duidelijk.
Ja.
Zeker als je met kleurtjes en zo.
Maar zo kunnen ze wel live volgen. Dat kunnen we met een screencast niet doen. Vroeger hadden we de camera niet en deden het via Skype for Business, omdat je dan op dat moment wel kan live meevolgen wat er gebeurt op je bureaublad. Maar daar was het gewoon, je kon bijna niet inbellen. En ik voelde in het begin van de les, dus je moest maken dat ik al klaar zat om mijn verzoek te accepteren als je online was. En dan zat je erin, en vooral dat er een klein beetje een storing was met de netwerkverbinding, was je eruit en kon je er niet meer in gaan. En dat was gewoon tegen het einde van de les, dat er niemand nog in mijn Skype-sessie. En had ik het voor niets gedaan, ja.
Dan kan je het niet alle twee ontvangen.
Ja, in principe wel. Ja.
Anders vinden ze het ook
Ja.
als
Maar je zeker voor dan thuis van kind bekijken. Het is wel nagaan, uw resolutie is beter.
Ja,
Dat is waar. Gaan we erin vliegen?
Ja.
We zijn een beetje te vroeg. Ik vind dat roest. Of jullie kunnen meer vragen stellen. So normally also the audio should work. And we start back in English. Yeah, looks fine. Okay, let's start. As you can see, the first slide is the introduction for the course unit. Because this is of course already your third course unit. It's going fast, very fast. But you didn't have the exam of the previous one yet, so this is next week, I think. They organize it in this way so that you have the opportunity to see each other before the exam. That's why they place the first contact moment of the next course unit before the exam. But we will change this for you. Let's start with having a look at the course site on Lego. As you can see on the course site on Lego, it has the same organization as before. So for the other course units, maybe I can try to say. So we have this home page. In the home page we have a general introduction, general objective for this course unit, which is called Comparative Genomic Analysis. Then you have the organization, so the different chapters that will be handled. And also, as you can see here, there is an assignment which is also part of the course material. So you should also have a look at the assignment that is available here. I already indicate from the beginning how the evaluation will happen. So for the evaluation, 100% of the evaluation is on the exam, which means that also the assignment will be evaluated on the exam. And here on the exam we have three parts. So normally you have two parts, closed book, open book part. Here it's a little bit different. So you have a first part with knowledge and insight, for which we use our laptop with lockdown browser. So I think that you use lockdown browser for, you will use lockdown browser for web technologies, which means that you're actually restricted to your exam and you cannot use any other applications on your computer. It is not possible to exit the exam, it is not possible to look something up, so you are restricted to your computer. Of course, this is more for theoretical questions, but of course also for insight questions. Then we have a second part, and the second part is more practical. So the second part will be an insight exam, where you can use everything on your laptop, besides the course material, so you cannot use the course material, but you can use all available tools, you can use all available databases and so on. So you can perform the exercises as you perform them during the lectures. So remember, part 1 and part 2 are both parts, don't use the course material, so it's not open book. You should know these two parts by heart. The third part is an open book exam, which also uses your laptop. This is for 20% of the exam, and this is for the assignment. So in the assignment, you will have to perform a task until the day of the exam, and there you will get some questions for the assignment. You don't have to hand in something, but I will ask questions about what the assignment is about. So for the assignment, you can use all information that you prepared for the assignment, some exercises that you made, some code that you made, and so on. And this is about analyzing a command line bioinformatics resource. So normally this is an assignment that we give for Linux, for the Linux course unit, but because you didn't have had any comparative genomic analysis yet, we cannot give the assignment there, because this is related to bioinformatics software. So here we will try to install some command line software that we can use for this course unit. An example here is we will install BLAST, we will install Crystal Omega, we will install Philip software. And for every software package, it is indicated what you should do. So you should look up how to install the NCBI BLAST Plus package, and here I indicate you can do this with DNF. So DNF we saw in Linux. Once you have installed this package, we will download the Swiss protein database from Uniprot. You can do this on the command line, you can do this by just selecting the file and downloading it in a web browser, of course. And next we want to create a BLAST database for this SwissProt file. Once we have installed our local database, you should be able to BLAST, and this we do with BLAST-P. Of course this is new, we will see this during the lectures. But you can use BLAST-P to BLAST a protein sequence against this database that you created. On the exam, you should be able to explain how you install this, how you install the database. You need to find these commands somewhere. Of course, there is some documentation available on the internet about this package, about how you perform these different tasks, but you should also be able to perform an exercise. For example, here I expect that you can use the BLAST-P program to BLAST a sequence against your local database in Linux. So this is all in Linux. The same for Crystal Omega. Crystal Omega is used to create a multiple sequence alignment, so these are the different tasks that we have. And then for Philip, Philip is installed in a different way. We cannot install this using the DNF command, so this is installed from source code. Of course, there is some documentation available on how to install an application from source code. Here I expect you to install the Philip package, and then I expect you to create a maximum likelihood phylogenetic tree starting from the multiple sequence alignment that you created in part 2. Of course, this may sound like Chinese because we haven't seen these different parts, but once we go through the course unit, we'll see that we can actually do this online or in a graphical utility, but you can also do this on the command line. This is what we want to show you, how you can do this on the command line. Of course, you are free to help each other using the discussion forum, so the discussions groups on the course site. It's an assignment, so I just want you to be able to perform these tasks for the exam, but it's not restricted to an individual assignment, so you can actually help each other to execute these tasks. So, what I want you to do, or possible exam questions, will be to explain how you solved these tasks from part 1 to part 3, both concerning the installation. For example, I can ask you, how did you install the Philip package? Then you have to give me the commands and the steps that you executed to install the Philip package. Another one is how to perform an exercise. For example, I give you some input sequences, and you need to construct a maximum likelihood phylogenetic tree. This can be a possible exercise. But for the assignment, so everything that you do here for the assignment is open book. So everything you prepared, you can bring to the exam and use on the exam to answer these questions. Is that clear? Okay. You can also find the example, so you can click this link, you can click this link. I made a couple of links just to remember you that you need to make the assignment so that you don't forget it, because it's 20% of the point. It's also here on top, so also here you have a button assignment. But I will change that button to exam on the exam day. So this will be a different button and then it will be gone. Besides that, of course, we have announcements. We have the course unit details, so the explanation about the different learning goals that are mentioned, that are handled during this course unit. But also the evaluation is discussed there, but this is of course also discussed below. And next we also have, of course, the organization of the contact moments. So I think that you already saw this for today, indicating when we can have these contact moments, the presentation for the contact moments, and what will be discussed on that contact moment. But of course also some explanation about the exam. For example, a link to the example exam. So this is an exam of a couple of years back. Actually, how I can ask you these questions. Of course, they will be solved on the computer and not on paper anymore. Next we have modules, and in the modules you will find the course material. So for this course, if we go to study material, there is a written syllabus in Word or in PDF, and we can actually see that the PowerPoint presentations are directly linked to the syllabus. So normally you should be able to find everything in the syllabus and in the PowerPoint presentations. Of course, the PowerPoint presentations can help you to add some clarification, some structure in the syllabus, to note what is important to actually have a look at. Besides that, also in the presentations there are some exercises which are not in the syllabus.
Is it like the course Linux that the exam is based on PowerPoints or the
Here
syllabus?
they are perfectly synced.
Ah, okay.
So normally I can base my questions on the syllabus or on the presentations, it would be the same. Yes. I will check that it is mentioned in both parts. Of course the exercises are not in the syllabus, are only in the PowerPoint presentations, so there you need to have a look at the PowerPoint presentations to be sure, to actually see these exercises. You can see that also the assignment is mentioned here, and here you can find the example exam. Next we have the different modules, which are the different chapters. So every chapter has a presentation, which you can have a look at. These are also the chapters from the syllabus, so you can find them there. And then we have some screencasts. These are only for the first modules, but I will add them for the other modules, where you can find some extra explanation about important parts of that chapter. For example, here I give you an overview of the databases that are important, what you can do with these databases, where you can find them, how you can search them, and so on. And also here, this is about genome browsers, how you can work with a genome browser. Very brief, of course, what I expect you to do, or be able to do. This is the same, for example, already for pairwise sequence alignment, because we will discuss these two topics today. Here you can have some screencasts about how to perform a pairwise sequence alignment using the NCDI BLAST utility, but also, for example, solutions to the exercises that are mentioned. These can be screencasts, but this can also be a document that is a solution to the exercise. You can, of course, first try the exercise and next have a look at the solution. I think this is a better way to do so. And I will also add this type of information for the other chapters. Is that clear? If you have questions, please ask me. Okay, perfect. Then we can have a look at the presentation for today. So today we will discuss two topics. First topic is an introduction to comparative genomic analysis. As you will see, most of the theoretical background that is in the syllabus I will not discuss during the contact moment. So here I will place the emphasis on a very brief theoretical explanation and on the exercises that you need to do for the exam. So this will be the focus of the contact moment, but there is of course a lot more information available that you should know in the syllabus. Especially for the first chapters, this will be, for example, about scoring matrices, and it's very important that you understand how they created these scoring matrices, how we can use these scoring matrices, and so on. But of course this is very theoretical, so I will not explain this during these contact moments. So we start with a general introduction to comparative genomic analysis. Then we'll have a look at the most important databases that we will use during this course unit. And next we'll have a look at a first step being pairwise sequence alignment. As you will see, the chapters are some kind of pipeline. We start with pairwise sequence alignment, then we use pairwise sequence alignment for database searching, then we can go to multiple sequence alignment, which is of course an extension, and you can use multiple sequence alignment for, and so on and so on. So it's actually some kind of pipeline that you can follow. For comparative genomic analysis, I think the subject says it all. We will have a look at molecular sequence data, most importantly sequence data that is available in databases. We will have a look at how we can compare the sequence data. Very brief, this all started a little bit in 1995. Was some kind of hallmark in comparative genomic analysis, where they sequenced the first genome of a free living organism. This is not that long ago, 1995. As you think about next generation sequencing was not yet developed then. So this was only started later. Next, of course, this was just a start. New sequencing technologies were emerging, and then they started to sequence thousands of other organisms. This can be on the level of DNA, so genomic DNA sequencing. This can also be on the level of, for example, RNA. So RNA sequencing is of course also involved. And you can even look at, for example, protein sequences. This is of course with different technologies. Mostly this will be mass spectrometry based, but also there you can sequence the proteins. it's not actual sequencing, but we can determine the sequence of a protein. And all this information needs to be stored. The first draft of the human genome was stored in books, was printed in a bookcase. This was one bookcase for one genome. Of course, if you sequence thousands of the genomes, you cannot store these in books. So this is not possible. And therefore they created these database structures. So they created structures. For us, these are web pages which we can search and we can retrieve sequences from. This is all in the cloud, meaning that we can actually share the information from one organization to another organization. And this is important for databases, is the possibility to share this information. Now, if we determine a sequence, we can store this sequence in one of the available databases. And the three most important databases that we have are actually distributed around the world. So we have, for nucleotide sequences, we have GenBank, which is located at NCBI, which is in the United States. Then we have the European Nucleotide Archive, which is on EBI, so the European Bioinformatics Institute. And also in Japan, there is an important database called the DNA Database of Japan. And this is also on the DNA database of Japan. So there they use the same name. So it's not like ENA, EBI, GenBank, NCBI. They use the same name for the database. Now, of course, we can choose where to submit our data. We can submit this on the European Nucleotide Archive, as we are in Europe. But you can also submit it in the United States, so at GenBank. This will not lead to a loss of information because on a daily basis they exchange their information. So all available information in one database is also available the next day in the other database. So you can find it everywhere. And this is of course done by an organization, so the INSDC, the International Nucleotide Sequence Database Collaboration, is organizing this exchange of information. We will focus for us on the first one, so we will focus on GenBank. We are in Europe, but we will not use the European version. Why is that? Because besides GenBank on NCBI, you have a lot of other databases available and also a lot of other tools available. For example, BLAST, which is Basic Local Alignment Search Tool, is a tool that we will use a lot and is developed at NCBI. And due to the connection of the databases with these tools and so on, we will restrict our use in this course unit to NCBI databases and NCBI tools. That's why we chose for NCBI. You could also do this, of course, at, for example, EBI. They also have sequence similarity search tools available, multiple sequence alignment tools available, and so on. So all these tools have some different flavors available on the web for the different institutes. We like to use GenBank to store our data. GenBank being the database with the most known nucleotide and sequence, we also say protein sequences, but this is called GenPact, so it's different from GenBank where we store nucleotide sequences, because here we have a file format called the flat file format where we have a lot of information for that sequence. Meaning that if we store a sequence in GenBank format, we don't only have the sequence itself, but we have bibliographic annotations, so references that are added, but also biological annotations. We can directly see which gene is this, which messenger RNAs are produced, where can we find the exons, and so on. So all this biological information is already added to GenBank. In GenBank, being the total database, we have a lot of information available. You can also see this in the graph, and you can also see that there is a new database called the Sequence Read Archive, which stores next generation sequencing data, which is actually exploding. Of course, the Sequence Read Archive contains this short read information, but is not well annotated. It's the raw data. If you want the actual sequences, full-length sequences, coding sequences, and so on, we go to GenBank. There we can find all this information. If you look at GenBank, we cannot just Google GenBank and go to the GenBank database, but we can actually access the GenBank database using the NCBI nucleotide database. If you go to NCBI nucleotide, and you search for a sequence, you open the record, you will see that this is in GenBank format. So nucleotide is only one of the possible entryways into the GenBank database. There are also others available. If you look at the format of this GenBank record, you can see that this starts with a header, and in this header we have general information. For example, we have a definition being, what are we looking at? This seems to be the AXO2 and REF7 genes, complete coding sequence from Saccharomyces cerevisiae. We also get an identifier being the accession number. So every sequence that you can find in a database has a unique identifier upon submission. And here you can see that the unique identifier for this sequence in the database is this U number. There are different formats for these sequence identifiers, which we call accession numbers. Accession numbers will be important during this course unit. You can also see a gene identifier, but they are removing the gene identifiers from the records. So this is something redundant and are not used anymore. Sometimes you can see that we have an accession number followed by a dot and a number. And this is the version number, meaning that if we publish our sequence in the database, we have version number 1. If we change something to our sequence or to our information, there is, for example, a sequencing error, and we correct it, then you get version number 2. And in this view, you will see that a lot of entries in the database will have different version numbers. You can also see the organism, even the taxonomic identification of the organism, and a lot of references, so bibliographic references from PubMed, that are linked to our sequence. Next, we have the features table. The features table starts with the term features and actually indicates biological information for your sequence. This is always indicated with position numbers. So for my sequence, I can find the coding sequence from position 1 to position 206. I can see that I have a second coding sequence from position 600 and so on to and so on. So here I can actually see the positions of different biological parts in my records. Of course, if you have a nucleotide sequence, not everything is coding. So not everything is translated into protein sequence, but you can have a look at that here. It is also useful if you want to save the sequence, for example, only the coding sequence, that you can select CDS. If you click on CDS, you will only get the coding sequence selected, and then you can copy the sequence to a new document. So that you don't have the full-length sequence, but only the coding sequence available. This is what we call the biological annotation. Next below, so the third part of my flat file format, of my genbank format, is the sequence itself. But here the sequence is numbered. So if we copy this, this is not very useful because we have numbered the sequences. We can also have these sequences in another format, and this other format we call FASTA format. And I don't know if it is available in the slides, the FASTA format. We will save it in FASTA format later. No, it's not available here. The FASTA format is a default format that we use for sequence data, meaning this is also plain text, as this is plain text. There's no makeup or anything available here. And in FASTA format we have a bigger than sign, followed by an identifier line, and on the next line we start with our sequence. We can interrupt our sequence with as many line endings as we want, so as many enters as we want, and this will continue until the next bigger than sign. Everything between two bigger than signs is our sequence. If this is on one line or on multiple lines, it doesn't matter. The identifier itself can be very specific, for example, for a database, but you can change this. So you can give it a name as you would like that. Once you enter, our software knows that this is the sequence that is starting. We save this as .fasta. .fasta or .fas or .fa. But we have to remove the .text extension. So this cannot be available in our file. So this is what we call the FASTA format. It's very simple. A multi-FASTA file has multiple sequences separated with these bigger than signs. And that's it. It cannot be more easy. If we are looking for sequence information, nucleotide sequences, we can start at NCBI nucleotide. Where can you find NCBI nucleotide? And this is something very easy for bioinformatics. You just go to Google, you type in NCBI nucleotide, and the first hit will be the NCBI nucleotide database. In the NCBI nucleotide database, you can search for sequence information. You can filter on taxonomy, you can filter on whatever you like. but you will have a lot of redundant information. For example, I'm looking for the rho kinase, and you can see that I already have a couple of chromosomes matching the rho kinase. I only want one record, so this can be a problem. If I ask you to give me a representative sequence for the rho kinase nucleotide sequence, and you have a lot of hits, this is not useful. And therefore there are some solutions which we will discuss a little bit later on how to find this representative sequence. If we are searching for protein sequences, we use the NCBI protein database. I don't think that this is that difficult. So we can search either of these databases. If we would select a record, we would be in the GenBank format. So you will see that this will always be in GenBank format with the header, the features, and the sequence itself. Another database that we have is where we can find cDNA data, but also express sequence tags. Express sequence tags are something special. This was eventually used in the beginning to determine gene expression of a gene. What they did is they had gene expression in the form of messenger RNA, and they started sequencing these messenger RNAs from the ends. And they had this single copy reads, short fragments that they could have from this messenger RNA. And determining, or by using the presence of these EST sequences, they used this for detecting gene expression. If they were able to detect, for example, 5 ESTs of this gene in a sample, they know that this was this much of expression. If they had 10 ESTs, this was double of expression of that gene. If you look in the database for expressed sequence tags, you can see that there are a lot of expressed sequence tags available, more than 8 million human expressed sequence tags, but there are only a couple, 22,000 genes, meaning that there is a lot of redundancy. We have a lot of different ESTs, which are of course different from each other, for the same gene. And this is what Unigene does. At Unigene, they cluster these EST sequences based on sequence similarity, so they are overlapping sequences, and they try to create one cluster, one EST cluster, using a lot of EST sequences for one gene. This also means, and this is different from the NCBI nucleotide and the NCBI protein database, if we search for a gene at the NCBI Unigene database, we should normally only find one record for an organism. And this is different at NCBI nucleotide, where you have a lot of different, you can have partial sequences, you can have mutant sequences, you can have a lot of different types of sequences, genomic DNA, contigs, and so on, that are all available in the database. And here we should normally only find one. For this one sequence, we can have a look at the available EST data, available cDNA data, but also, for example, at EST expression profiles, where we can have a look at approximate expression patterns of my gene in different conditions. This can be disease states, this can also be body sites, developmental states, and so on. So this information we can all find in the Unigene database. As indicated before, it is not easy for us, if we want to find a representative sequence. Of course, there are a lot of different variants available in the population, but we want to find one representative sequence of a gene or from a transcript to search the NCBI nucleotide database. There's a lot of redundant information available, and if we only want the representative sequences, we can start our search at NCBI gene. NCBI gene is a little bit the same as NCBI Unigene, saying that they remove the redundancy and try to cluster the information for a specific locus. Meaning that if we look at the genome, we have a locus, a position on the genome, a fragment, and here they try to actually combine all information for such a locus in one page. So if I search for the rho kinase, I normally should only be able to find one rho kinase locus in humans. So I can directly find my rho kinase in humans. On that page, a lot of information, locus-specific information, will be available. And we'll have a look at that. You will see that there is, even for that page, a table of contents, so that you can navigate to different parts of the web page in a fast way, without actually having to scroll the whole page. What else do we have on NCBI? is the taxonomy browser. The taxonomy browser provides you with an overview and an organization, so a hierarchical, I think it's a difficult word, some kind of classification system for organisms in the database. Why can we use the taxonomy browser? This is especially useful for us when we are looking for information for a specific organism. For example, I'm interested in genes in HIV-1. Then I don't go to NCBI nucleotide and filter on HIV-1, but I can start by looking for HIV-1 and then I can go to genes. And the filter for HIV-1 will be automatically applied in our database. There is a connection to every other database starting from the taxonomy browser. So if you start at the taxonomy browser, you can go to protein, you can go to nucleotide, you can go to whatever you want, NCBI gene, EST data, and so on. So you can always start for an organism at the taxonomy browser and then look further to the other available information. Of course, if you're using human, most hits will be for human on top of the results list, as this is used the most. So here for human, this is not used a lot, the taxonomy browser, but if you're working with other organisms that are not that well known or less known, then you can start at the taxonomy browser and have a look at the available information for that organism. Other databases that are available at NCBI are, for example, PubMed, which is for biomedical literature. We also have the Entree system, but this is actually the search system that is used at NCBI. So if we search in a database, we actually use the Entree system to search that database. This is something special. We also have BLAST, so the basic local alignment search tool available there. We also have OMIM, which actually tries to link genetic variation to human diseases. So if a disease has a genetic cause, then we can find it, if it is known, in OMIM. Also, if we search for a gene in OMIM, we can have a look at the phenotypes, the diseases that are associated with that gene. This we can find in OMIM. Before, this was not OMIM, this was just called MIM because these were books. But because of the information that is updated a lot, they made an online version of MIM, called OMIM, the online Mendelian inheritance in MIM. As indicated before, every sequence, every type of information that is submitted to one of the biological databases gets a unique identifier. This unique identifier we call an accession number. It is not possible that two records will get the same identifier, so it's not possible to have the same accession number. This is not possible. If you want to save information, for example, you have found a sequence, and you don't want to save the sequence itself, but you want to retrieve that information later, you can just note the accession number, and you can use the accession number to find the information back later. Also, for searching sequences, multiple sequences, we can easily use the accession number to have sequence information for multiple sequences at the same time. Besides the simple accession numbers that we use, there is a project which was also, I already indicated, because of the redundancy in the databases, and this is called the RefSec project. Because there is a lot of variation in our genome, a lot of genetic variation, we have different alleles that are possible within the population. They tried to create one representative sequence for every gene, for every protein, and so on. This representative sequence should be the consensus sequence, meaning the sequence with the highest frequency in the population. If we have a mutation, we should compare the mutation with that reference sequence. They had this project called the RefSec project to actually create these reference sequences, and we also have these types of identifiers. So these accession numbers are in a special format. You can find this on the next slide. These are the formats that are used, and they indicate that we have a reference sequence. So these are RefSec identifiers that we use here. The goal is that for every gene or every protein, we only have one identifier. So it's not possible to find multiple RefSec identifiers for the same gene. This is not possible. But there's one difference here, and this is for splice variants. So this is a little bit of biology. If we have a genomic locus, so a position on the genome, a gene on the genome, we can only have one RefSec identifier for that gene. because there's only one locus containing that gene. If we have transcription into transcripts, RNA, we can have multiple transcripts variants due to alternative splicing. These are actually different versions of the transcript that can come from one genomic locus. And there you can actually have, for the same gene, multiple identifiers, because every transcript variant will have a different identifier. So this is possible. One gene, multiple identifiers, because we have alternative splicing and thus we have splice variants available. Of course, these splice variants will lead into different isoforms. So on the protein level, when we translate these splice variants, we have different isoforms. We call them isoforms. And these can be identical. If they are translated into the same protein, they are identical. But also they will have a different RefSec identifier because they come from a different transcript. So there we can have identical proteins with different RefSec accession numbers because they come from a different transcript. These accession formats are very easy because they indicate what type of data that we use. We will focus the most on these two, being messenger RNA and M numbers. So the M is for messenger RNA, protein sequences and P numbers, we will distinguish. If we have RNA that is not messenger RNA, this is our NR numbers. So we can already see by looking at the accession number what type of data we have here. If you look at, for example, complete genomes, you will see that these complete genomes will have an NC number. If we look at, for example, genomic DNA sequences, so the genomic DNA sequence of a gene, this will have an NC number, but a range, indicating that in position 1 to position 500, for example, we have that gene on that genomic sequence. So there they have the same ID number, but a different range within that ID number. Besides the sequence database, there's also something that we call genome browsers. So it's also possible to have a look at the genomic context of a gene, to have a look at flanking genes, at upstream or downstream sequences of a gene, and so on. And this we can do by using a genome browser. There are a lot of genome browsers available. So we have the genome data viewer, which was previously called the map viewer. You will maybe not know this, but last year this was called the NCBI map viewer. This year this is called the NCBI genome data viewer. This is the genome browser from NCBI. We also have a genome browser on the UCSC, so the University of California Santa Cruz website, which is the UCSC genome browser. And we also have a genome browser at Ensemble. So these are the three most important genome browsers that we have. And here we are actually moving away from NCBI because we will use the UCSC genome browser for our exercises and not the map viewer. This has some simple reasons, because this is a very good genome browser. This is one. But also because if you go to, for example, next generation sequencing, it is possible to add your own tracks to this genome browser. So you can add your own data, or you can map your own data or visualize your data on this genome browser by adding new tracks. Maybe in other course units you will be able to use this functionality for your data. Or you can do this at your work placement or somewhere else. There you can actually add your data to the UCSC genome browser. And that's why we chose the second one. So the UCSC genome browser is a very good choice of genome browser. It's well annotated, there's a lot of information available there. It's more than a genome browser. You should of course indicate when you use a genome browser which assembly, which version of the genome that you are looking at, and it's also possible here to add your own data. And I think that I've said enough at this moment. So we can start with some exercises. I think this will be the most important part, that I show you where you can find this information, how you can get this information, and so on. So these are simple exercises. I am looking at a gene, FMR1. It's a human gene. Most of the examples that we use will be human. Of course, on the exam we will not use human, so you should be able to look up information from another organism. And there you will see that I want to find the messenger RNA sequence of this gene, but also the protein sequence, and I want to save this in FASTA format. So this doesn't sound that difficult, just save the messenger RNA and the protein sequence of FML1. Next, determine the locus of FML1. So where can I find this gene on the genome? They determine locus, loci, I think it is, and if you have multiple, it's loci, I think. And they indicated these loci numbers by using Ginza staining. So this was actually in the wet lab. They determined these band patterns on the chromosomes, and here we can actually distinguish the locus, so the position of genes on the genome. Next, give me the RefSec identifier of this gene, not messenger RNA, not protein, this gene on the genome. Look up the amount of EST and messenger RNA sequences that are available for FML1. Is there a disease related to FML1? But also, how many human clusters can we find in the Unigene database for, not for FML1, just in general, how many Unigene clusters can we find? Think we should do this together? But you can answer. Google. So I'm looking for the messenger RNA and the protein sequence of human FML1 in FASTA format. Which database, NCBI database, can I use? Let's start with the first one, so the messenger RNA sequence. Where can we find nucleotide sequences at NCBI? NCBI nucleotide. You can see the first hit is actually NCBI nucleotide. So I select NCBI nucleotide. And at NCBI nucleotide, I search for FML1, human messenger RNA. You get some kind of pop-up. And then if you look, there are actually 700 possible sequences in the nucleotide database that match our term. You can see that the first one are Arabidopsis, so my human did not work. But of course we can restrict our search here to Homo sapiens. Then we have 172 possible nucleotide sequences. And then you can see that our first hits are chromosomes. This is not what I'm looking for, but of course these chromosomes will link to messenger RNA and so on, so these databases are connected. So here we have a lot of redundant information. How can we solve this redundant information if you don't want to have redundant information but consensus, representative sequences? Where do we start? It's not at NCBI nucleotide, but at this is for cDNA and EST data and expression data. NCBI gene. So if you look for sequence information and you want representative sequences, we always start at NCBI gene. Here, NCBI gene. If I search FML1, enter, in 95% of the cases, your first hit will be the correct one. Because they list Homo sapiens on top as first hits most of the times. And you can see that at the top list, you only have one hit for Homo sapiens, which is called FML1, and the long name. And here you can already see the chromosome sequence, the RefSec identifier for that chromosome sequence, and you can see that this is located on chromosome X, so in this record with these positions. There you can find that gene. You can also see that there is a link to MIM, Mendelian inheritance in man. If I select my gene, you can see, and you have to wait a little bit because it's still loading, so this is a little bit slower. And if you look at my scroll bar, this is very small, meaning that there is a lot of information on this page. Really a lot of information. And therefore we can use this table of content. The table of content allows you to jump to a specific position on the web page. I am looking for a representative messenger RNA and protein sequence. And if I look here, so summary, genomic, and so on, and so on, and so on, and so on, here I can see NCBI reference sequences. So if I'm looking for a sequence, I go to NCBI gene, search for my gene. Most of the times this will be the first hit. And there on that gene page, I can go to NCBI reference sequences. For these NCBI reference sequences, I can see that they make a distinction between genomic DNA, messenger RNA and proteins, and that's it. And RNA, even RNA sequences here. So which are not messenger RNA sequences. So for the genomic DNA, I should only be able to find one hit, one accession number. And this is this NG number, which is indicated the RefSec gene. For my messenger RNA and proteins, I have 1, 2, 3, 4, 5 different transcripts. Meaning that we have alternative splicing, which results in 5 different transcript variants for this gene. Every transcript variant results in the translation of a protein. So we will also have 5 different protein isoforms. So these are the messenger RNA records, these are the protein records. If we select this, we will be redirected to NCBI nucleotide, to the GenBank record of the reference sequence. If we select this, we will be redirected to NCBI protein, to the protein record of the sequence. You also have some information about this variant. So this variant lacks an alternate segment and uses a different splice site at the 3' coding region, which shifts the reading frame compared to variant ISO 1, and so on. If I have multiple variants and multiple proteins, and I ask you to give me a representative sequence, most of the times we use the longest one. So we search for the longest transcript variant. This lacks an alternate segment and is shorter when compared to isoform 1, so I will look for isoform 1. Isoform 1 is here, it's number 5. It's not logical, but it's number 5. It's called isoform 1. This transcript, this variant, represents the longest transcript and it encodes the longest protein. So if I look for a representative, only one sequence, I will use this one. We have 2 accession numbers, one for the messenger RNA, one for the protein. I want a messenger RNA, so I will select the NM number. I do this with Control-click, so that this opens in a new tab. This is, of course, you can choose this, but just to give you some help, Control-click. This will be the messenger RNA sequence. I can also already click the protein sequence, Control-click the NP number. This will open the protein record. As you can see, if I open the messenger RNA record, this is in GenBank format, and you can indicate this here. So this is GenBank format. If you want another format, you can select this and you can choose another format. For example, the FASTA format is already present here. So if I only want to sequence in FASTA format, I can select FASTA here. This will still be on a web page. So you will still have a web page with the FASTA sequence in the middle. If you only want to sequence without the web page, you can choose the FASTA text. This will only give you the FASTA sequence without the web page. So this is possible. As indicated, I have my accession number, which is a RefSec identifier, some general information. A little bit below, I have my features table, so this starts here, indicating the position. So in total, we have 4,411 nucleotides or bases in this sequence. We have our gene, which is of course the sum of that, and then we have our exon, the first exon, I go a little bit further. We have our coding sequence which is indicated, and this coding sequence is of course the region of my transcript that is translated into protein. For analysis, it is often good to restrict or search to the coding sequence, because this will be more conserved than the untranslated regions at the ends of my transcript, which are less conserved than the coding sequence. If I want this in FASTA format, I can select FASTA. You can do this. Then here you have the FASTA sequence. So it starts with the bigger than sign until the end. Up. This is my FASTA sequence. I can copy this to a text editor, a plain text editor. If I, for example, only want the coding sequence, I can select the CDS tag. So here we have CDS. If I click the CDS tag, it will only select the coding sequence. And if you now want the coding sequence in FASTA format, you can select the FASTA button below. And now you will see, this is also indicated here, that we only have that region of my total transcript sequence which has the coding sequence, and this is also indicated here. And this I can save. If I want to save this in FASTA format, there are some Mac users, we need a plain text editor. I think this is called text editor on Mac. On a Dutch Windows computer, this is called Kladblok. This is plain text. It's not possible to paste something in bold or italic or in another way. This is just plain text. Here, we can copy our sequence. And I will show this for you. So I copy my sequence, Ctrl C. I go to my Kladblok document, my plain text document, and I paste my sequence here. And if I now want to save this as FASTA, I go to File, Save As. And with File, Save As, let me create a new directory at home. With File Save As, I can add the name, messenger RNA of FMF1 dot FASTA. So I add the FASTA extension myself, but if I save it now, it will also add the .text extension. And some software will not recognize this as a FASTA document if you add the .text extension. And therefore we need to remove this .text. It's difficult. You have to try this a couple of times. How can we remove the .text? Just by saying that we want to save this as all documents. And if we now save it, it will recognize the .fasta and not the .text. And it will not add the .text. And this is important for us, that we remove the .text. If we want to create a multi-FASTA file, then we go to the last line, we enter, and we start a new bigger than sign with an identifier, and then we start our sequence on the next line. Of course, if you create a multi-FASTA file, don't combine nucleotide with protein sequences. So these we split up. You can have multi-FASTA files of nucleotide sequences, multi-FASTA files of protein sequences, but we don't combine them. Let's do the same for the protein. So I go to my second tab, which is the protein record. You can see this is my NP number. This is not called GenBank. It's actually the same format, but this is called GenPept. This is in the protein database. And also here I can go to specific regions that I have here. Of course there's no coding sequence because my coding sequence is everything, because it is already translated. If I want this in FASTA format, let me select the home button. Again, here I have FASTA. And I can copy my protein sequence to a new plain text editor. I do not say Kladblok document. And here I can paste it, and this I can save again. So save as. and I can call this protein of FMR1 dot FASTA. You can add dot FASTA, you can add dot FAS, you can add dot FA. This is all recognized. So these are different extensions, different suffixes that you can use for the FASTA file format. Most of the times we use FASTA. As you can see from my previous one, this is indicated with FASTA file type and not .text. So FASTA file type, and then we can save this. Next question, determine the chromosomal locus. You can find this on multiple pages. So everywhere where you search for FMR1, you will be able to find the chromosomal locus. Of course, for us, maybe it is most useful to actually look at the NCBI gene page. So I'll remove the protein, remove the messenger RNA, and here I am on the NCBI gene webpage. I go up with the home button on my keyboard, then I am on top, and then I can have a look. One of these first titles will actually contain my locus number. If I come down to genomic context, I can see my locus. It's indicated here that I am on chromosome X, on arm Q. Is this the long arm or the short arm of my chromosome? Long arm, yes, because the P is the short arm from petit in French. And then we have locus 27.3. So this is my locus X chromosome, long arm 27.3. We have for this gene 17 exons. Next question. What is the RefSec identifier of the FMR1 gene? I can find this with NCBI reference sequences. So I go back to my NCBI reference sequences. I am interested in the RefSec identifier of the FMR1 gene. So I look at genomic information, the RefSec gene. So this is my RefSec identifier of the FMR1 gene. If you want to go to the GenBank record, I can select GenBank. And then you will have the GenBank record for that one gene. Next, look up the amount of EST and messenger RNA sequences for FMR1 in the NCBI database. If I'm talking about EST sequences, where will I look? Unigene. So here we look at Unigene. So I can start a new tab. I search for NCBI Unigene. My first hit is the best one. At Unigene, this is also focused on the locus, meaning that we don't have redundant information. This is a little bit the same as for NCBI gene. If I search for FMR1, I should normally only find one record for FMR1 in humans. As you can see, in 95% of the cases, the first hit is the correct hit. I have my Fragile X mental retardation 1 record, FMR1. And here you can see that for this gene, we have 255 EST sequences and 133 cDNA libraries available. Of course, cDNA is just the complementary DNA of my messenger RNA. So this is the available sequences that we have here. You can also see, because we have 7 different reference sequences, transcript variants, we do not get the accession numbers here, but sometimes you get the accession numbers here. If you want to have some information about the expression of this FMR1 gene, you can have a look at the EST profile. So these are approximate expression patterns based on EST, express sequence tagged sequences. And here you can see the expression by body site. We can have a look at the expression by health state and at the expression by developmental stage. the expression itself is indicated in transcripts per million. So from a sample, they isolate all ESTs, and then they count the number of the ESTs on the total that are from your gene. And these are transcripts per million. So if we would have 1 million transcripts in total, here we would have 98 transcripts that are from FMR1. If you would have 1 million transcripts in total. And in this way we can indicate the amount of gene expression. Of course, this is also indicated with some kind of dot, and the darker the dot, the more gene expression you have. Which inheritable disease is related to FMR1? The almost last question. Where can I find this information? Do we know a database? Online Mendelian Inheritance in MAM, so OMIM. So I can search for OMIM. OMIM has two portals, so two possibilities to search OMIM. You have one available at NCBI and you have one on its own web address. They are the same. I prefer the web layout of omim.org. There's only one thing that they ask you, and this is to donate. You are of course free to donate. Here we can search for FMR1, enter, and we can find our FMR1 gene as a first hit. For this FMR1 gene, I can have a look at the gene-phenotype relationship. As this database is for diseases, if you talk about phenotypes, these are of course diseases. So a disease phenotype. Not just strong arms and long legs. So actually diseases. And here you can see that there is a link between FMR1 and these diseases, where you can find, of course, the Fragile X syndrome. But if you look at the name, Fragile X mental retardation protein, this was already indicated in its name that it would have something to do with this disease. Last question here, how many human clusters does the Unigene database contain? Do I go to Unigene and search for human, or if you want species-specific information, where can we start the best? If this would, for example, not be human but another organism?
The taxonomy browser?
Yes, the taxonomy browser. So let's go to the taxonomy browser. Where can I find it? Google. You are allowed to use Google on the exam. So we search for the taxonomy browser. In the taxonomy browser you can search on trivial names, human. You can also use the correct names, Homo sapiens and so on. But for humans there's also a short link here. Let me start with, for example, Homo sapiens. There's a dollar sign. Homo sapiens. And here you can find the taxonomic names of Homo sapiens. You can see that we have humans, Homo sapiens, as one organism, but we also have two subdivisions. We can have Homo sapiens neanderthalensis, yes, or Denisova. These two are extinct species from humans. But they are also, or there is some sequence data available for these species in the database. Of course, if I'm looking at a modern human, I am interested in this one. So I can select the first one. The other two are subspecies, as we call them. Here, for an organism, we get some general information, and we get a table on the right. And it's this table that allows you to link my organism to a database. If we now select, for example, nucleotide, and we select this, we will be redirected to NCBI nucleotide using the filter for the organism. Here we have the different databases that are available. We can see the number of records in these databases, but there's a distinction between subtree links and direct links. Most of the times you will see that the number of subtree links is bigger than or equal to the number of direct links. What can I find in this column, and what can I find in the second column? What does that mean? Subtree links and direct links. Some ideas? If I go back, I selected Homo sapiens. This is my direct link. If I include subtree links, I will not only find records for Homo sapiens, but also for all subdivisions. So, if I select, and then I will go back to my next page, if I select here, I will also find Neanderthal and Denisova records. If I select the second, sorry, this is the third column, I can only find Homo sapiens which are not Neanderthalensis, not Denisova. And that is the difference. How many human clusters can you find in the Unigene database? As you can see, and therefore I searched for Unigene, you can see, and this is the same, there are 130,000 approximately Unigene clusters available in the database. You can only find one genome. So there's only one human genome. If you look at the number of nucleotide sequence records that are available, or SNP records, so single nucleotide polymorphisms, mutations in our DNA, you can see that these are very high numbers. If you would, for example, search for Neanderthalensis, this would be very limited, the list. There's only a little bit of information available at NCBI for these organisms. Let's go to the next exercise. This is about the genome browser, so determine the location of the human ctNNA3 and FMR1 genes, and which of both genes is located in a region with a lot of other genes. As indicated, if we're looking at the location of genes, at the genomic context of genes, we use genome browsers to have a look at flanking genes, at their positions on the genome, and so on. So therefore, I will start with going to the UCSC genome browser. Also here, we can start at Google, and we can search for UCSC genome browser. The first one is the correct one, so the UCSC genome browser home. And here on the UCSC genome browser home, we can select the term genome browser on top. So here we have genomes, genome browser, tools, mirrors, downloads, and so on. And here we just select the name genome browser. So not configure, not track, just the term genome browser. And then we will be redirected to our genome browser. Where are we now? Now we are on chromosome 16, locus P13.3. Here you can see an ideogram layout of chromosome 16, and the red bar indicates where we are. Now this is a bar because we are zoomed in. If we zoom out on the genome, this will become a box indicating the view of your genome that you're looking at. You can try this. We have controls, move to the left, move to the right. We can zoom in, we can zoom out. We can zoom until the base level. If I select base, we can actually see the sequence, see the translation that we have. This is all. This can be another gene. So it should not be this perfect gene. And here I can actually search for a gene. I will start with searching for ctNNA3. I type in ctNNA3, but I do not press enter, because this will give me some drop-down list with possibilities. And here I can find my ctNNA3 record, so that I have the correct one, and this will not be a keyword search. If I have selected the correct one, I select go. And now I will be redirected to the position of ctNNA3 on the human genome. As you can see, we are on chromosome 10, locus Q21.3, but if I ask you for the exact location, I ask you for this information. This information. This is the exact position of my gene on the genome. Of course, you should know that there are multiple different versions of the genome. There are different projects that try to solve the human genome, and they all have their own version. Meaning that this depends on the version that you are looking at. If there is a new release every couple of years, there is a new release of the human genome, they update the information that is available. These positions can change. If we go to these positions, this may or may not be the CTNNA3 gene. Therefore, it is important to note on which version that you are looking at. And this you can find on the top right. So this is the Genome Reference Consortium Homo sapiens 38 version. We call this an assembly. So an assembly is a version of the human genome. This also has a short abbreviation called HG38. It's the same. You can use one version or the other one. It's the same version of the human genome. So if I ask for the exact position, you give me the numbers, chromosome 10, position, and then there's a lot of numbers. But you have to indicate that this is on assembly GRCH38. This is important. There are previous versions, GRCH37, and other numbers, and the positions can change. So here I search for CTNNA3, I can find it here. If we are interested in NCBI RefSec genes, you can see this is the NCBI RefSec genes, and there are different. There are here 1, 2, 3 versions. A short one and 2 longer transcript variants. You can see that the direction is indicated with the arrows, so the direction is from right to left. So this is the 5 accent, this is the 3 accent. The blocks are my exons, and the lines are my introns. So these are introns, and this is a block, is an exon. If this is a larger block, so this is large, this is actually translated. If this is a smaller block, as in the beginning here, this is not that wide, this is untranslated. So this is not translated into protein. Next, I'll duplicate my window. Luckily that my circle stays the same. And I can search for FMR1. I wait and I select the correct one. I press go. And now I want to have a look at the genes that are in the neighborhood of my genes. You can see that there are a lot of more transcript variants available. This is possible, but this of course has no effect on my context that I have here. I look at the NCBI reference sequence track. You can hide the other tracks. For example, here we have the GAN code track. If you don't want to see it, we can do right mouse button hide and then it's gone. You can add tracks below by changing the view of them from hide to show or dense or full. In this way you can actually see them and refresh your view. If I want to see the context of them, I can zoom out, for example 10 times. And I can also do this for CTNNA3. Zoom out 10 times. I think this is my NCBI reference sequences. Maybe it's too much. I can see all these small lines indicating all genes. Also for FMR1, I can see only a couple, see my FMR1, but nothing else that is available here. Maybe I can change the view to pack. Yeah, I can change my view to pack. And then I see it a little bit better. So if you look at the information that is available here, now it's the same. Or not? Yes, it has changed my view. So this is for FMR1. If I do this for CTNNA3, NNA3, but it's possible that this will synchronize. I'm too fast. CTNNA3. And I zoom out 10 times. You can see that there are a lot more genes available. I think this is my gen code, so I will hide this. this is my RefSec, there are a lot more genes available here than there are available for FML1. Is this correct? That there are a lot of more genes in the context flanking my CTNNA3 than there are genes flanking FML1? If you look at these views, this is correct, but have a look here. My scale is 1 megabyte. Here, my scale is 5 megabyte. So I'm looking at a different zoom level. Here I am more zoomed out, and therefore I see more results. So it is important, if you try to compare these views, that you're actually looking at the same magnification. En nu hopen dat hij niet uitvalt in die 2 seconden. Dat zou stom zijn. So here we have to make sure that we're looking at the same magnification of our region. And then you will see that this is actually quite the same for both genes. So this was the first part. Let's start with the second part, being pairwise sequence alignment. And I'll try to remove my circle from the screen. But it's not easy because they've changed the organization of the classroom. So in the second part we'll have a look at pairwise sequence alignment. We're looking at pairwise sequence alignment because we're going to use this, for example, when we search a database for a related sequence. So the question that we have here for pairwise sequence alignment is we ask ourselves, are 2 sequences related? If we have 2 nucleotide sequences, or if we have 2 protein sequences, we want to know if they are related, meaning that they come from the same ancestral sequence. So once upon a time in history, this was one sequence, and today we are looking at 2 different sequences, but they were originated from the same one. In this way, we ask ourselves the question, are they homologous? Meaning, do they have the same ancestral sequence? We can also ask this question, this relatedness question, to have a look at the function. If we know the function of one sequence, and we don't know the function of a second one, but we see that they are similar, we can actually try to determine that function. If we compare our sequences, we do this by protein sequence alignment. So if we want to find related sequence, we do this most of the times on the protein level, because we have more information available on protein level. There is less redundancy due to redundancy in the genetic code. And in this way we can actually detect, are you homologous or not? So are you related or not? So if they are homologous, they share a common ancestor. Normally we can determine this by having a look at the 3D structure of our proteins. If the 3D structure looks the same, then we know that they are derived from the same common ancestor. Of course, we don't always have 3D structure information available, and that's why we look at percent identity on the sequence level. So we use percent identity to determine the relationship between sequences. If I ask the question, are 2 sequences homologous, you cannot answer with 50%. So homologous, the definition is they are derived from the same ancestral sequence, meaning they are or they are not. So this is a yes-no question. You cannot answer they are 50 or 30 or 70% homologous. This does not exist. To determine homology, we can use percent identity or percent similarity. And this is something quantitative. So here you can actually have a number. They share 80% identity. They have 80% identical residues. So this is possible. Just to give you an example, if we compare human myoglobin with human beta globin, and we look at the 3D structures, we can definitely see that these 3D structures look the same. They are homologous, they are derived from the same ancestral sequence. But if we look at the percent identity, they only share 26% identical residues. So for a pairwise sequence alignment tool, it's very hard to actually distinguish or to see that they are homologous sequences. Next, between homologous sequences, we make a distinction between what we call orthologous and paralogous sequences. Orthologs are homologous, which are due to a speciation event. Which means if I, for example, look at myoglobin, and I look at myoglobin in humans, and I look at myoglobin in rat, this is the same gene, but just in another organism. There they are different, two different sequences, but once upon a time there was one ancestral organism, some kind of rat human, which had only one gene. Today, these are two different organisms, but it represents the same gene. This is due to speciation event. There was a speciation, we had the ancestor of the human with the rat, and then they split up in evolution. It's the same gene, but in different organisms. Here we know that the function should be the same. So normally this gene would have the same function in both organisms. Besides orthologous, we also have paralogous sequences. Paralogous sequences are not due to a speciation event, which means if we look in one organism, we can find paralogous sequences, and this is mainly due to duplication events. It means that once upon a time we had one, it's like a fairy tale, we had one sequence, then there was some kind of genomic alteration, we had a duplication of that genomic sequences, and then we suddenly had two sequences representing the same gene. But because they are duplicated, the evolution on the one sequence and the evolution on the other sequence differs. And they can start to change from each other. And these are called paralogous sequences. Normally they should be similar. If this was a recent event, they can be 100% similar. For example, human alpha-1 and alpha-2 globin are from a recent in terms of evolution is not that recent, but recent duplication event. They are 100% identical, but still similar function with different properties. How can the properties of human alpha-1 globin and human alpha-2 globin differ? They are 100% identical, but still they have different properties. What do I mean with different properties? No, they are identical. If we meet them in the cell, we cannot distinguish them. But still they have different properties.
Different place of expression. Different place of expression.
Yes. This can be due to a different place of expression. One is expressed in my knee, the other one in my liver. And therefore they have different properties. This can be time, place, but also amount of expression. This can be that alpha-1 has a very high expression and alpha-2 has a very low expression. So these are different possibilities that we have. This can also, for example, be that alpha-1 is expressed in healthy individuals and alpha-2 is expressed in sick individuals. So also a different physiological state is possible, although they are actually the same. Now, not all paralogous sequences are 100% identical. If you look at, for example, I'll go one slide back, human myoglobin and beta globin are in the same organism. Once upon a time, this was one sequence. There was a duplication event a long time ago, and today we cannot recognize them anymore as homologous. We can, but almost not. So it's not that all paralogous sequences are actually 100% identical. In later chapters we will have a look at how to construct phylogenetic trees, so to determine evolutionary relationship between sequences, here you should actually think that on the ends we have a sequence. A chicken sequence, a dog sequence, a mouse, a rat, zebra, horse, sperm whale, and so on. So these are all sequences at the ends, and the relationship between these sequences is depicted with this tree. So the two most closely related sequences that we have here would be the horse and the zebra, because they are at the same line. The more or the bigger the branch length that we have horizontally, the further the distance from these two sequences from each other. So for example, from sperm whale we have a very long distance to chicken, because we have this amount of evolution. So we can indicate the branch length as an amount of evolution, a number of changes that we have. Here you can see that we all have different organisms, but every sequence actually represents the same gene, being myoglobin. Therefore, because they are all in different organisms, these are orthologous sequences of myoglobin, all in a different organism. If we look at the next one, here we can see all different types of globins. So as you will also see in the examples in the course unit, there are a lot of examples using globins because there are a lot of globins available. A lot of groups of globins. We have the alpha globins, we have the beta globins, we have myo, neuro, cytoglobin, and so on. If we all place them in a phylogenetic tree to determine their relationship, for example, beta globin we have here, and then we have myoglobin which is there. They are only 26% identical. If you have alpha 1 and alpha 2, they are 100% identical. We can depict their relationship, but this is all in one organism. We can construct this tree for humans, we can construct this tree for mice, we can construct this tree for other organisms. So therefore, because these are all duplication events on these nodes, we call this paralogous sequences. So this is the relationship between these homologous sequences, they are paralogous. So if we want to determine the relationship between two sequences, for example, this one and this one, we just trace back to their node, their event where they split up, and we try to distinguish what is this event. Is this a duplication event or is this a speciation event? If I can find, for example here, and I split up my tree in two parts, part 1, part 2, if I cannot find the same species on one side and on the other side, this is a speciation event. So if all species on one side are different from the species on the other side, this is a speciation event. If I can find the same species on multiple sides, this is a duplication event, because the species is still available on both sides. Then we duplicate our DNA. So this is how we can actually have a look at these trees. Therefore, my next question, we know that human alpha and beta globin are paralogous sequences, meaning we are in the same organism but have two different versions due to duplication event. The same is for mouse alpha and beta globin are paralogous. If we would only look, for example, at the alpha globins from human and mouse, we would say that these two are orthologous sequences. Now, if I compare human alpha with mouse beta, what is the relationship of these sequences? Are they, of course they are homologous, we already know that. Are they orthologous or are they paralogous sequences? They can be both. So depending on the events that happened during evolution, they can be both. So it's possible that we first have a duplication event, and in the duplication event, we split up the alpha from the beta. Next, we have a speciation event, creating the human alpha and the mouse alpha, the human beta and the mouse beta. Here, this is due to a duplication event. So in this situation, duplication, these are paralogous sequences. If we first have a speciation event, I need my wiper. We don't know if we call this a wiper. So if we first have a speciation event, human, mouse, and then we have a duplication event, human, alpha, human, beta, mouse, alpha, mouse, beta, then these are orthologous sequences. But of course this means that this duplication event and this duplication event happens in two separate organisms, and thus, if they happen in separate organisms, this is very unlikely that the duplication event happens in two organisms at almost the same time, and thus results in alpha, beta. Well, of course, it is possible. That's possible. So depending on the order of the speciation and duplication event, we can determine the relationship if they are orthologous or paralogous sequences. How can we compare them? How can we determine the relatedness? By using pairwise sequence alignment, and the easiest way to do this is by using the NCBI BLAST pairwise alignment utility. This is the easiest way. I should say that there are some possibilities at EBI, so the European Bioinformatics Institute, some sequence similarity tools to perform a global or local pairwise sequence alignment, which are not available at NCBI. You can do this for protein sequences, for nucleotide sequences. You can even use the accession numbers, so the identifiers from the database, as input for your comparison. So you don't have to paste the whole sequence or upload a file. You can just use the accession numbers and it will fetch the sequence itself. We can add other different parameters, for example scoring matrices and so on. We can all change this to adjust the way our tool works. In pairwise sequence alignment, we actually try to create or to align two sequences as good as possible, and this means that we have the maximum amount of identical residues. We try to have the maximum number of identical residues. Of course, we can score these alignments, and we try to have the alignment with the highest score. This is what we try to do. So we determine degree of similarity, or we can determine homology. By using pairwise sequence alignment, we just try to answer the question, are they related or not? Of course, there's also some statistics behind these pairwise sequence alignments. They try to compare this with what if they are not related, if these are random sequences, can you get this type of alignment score? And this is also given in the form of an E-value, which represents some kind of P-value. So the probability that they are homologous, but we don't see them as homologous. So this is also given here, but we will see that later. And this was already stated before. If we create a pairwise sequence alignment, we try to find the best alignment. For short sequences, this is possible, and especially for pairwise sequence alignment, it is actually possible to get the best possible alignment. And if we have an alignment, you can score this alignment. We score this alignment by having a look at matches, by having a look at mismatches, and by giving scores for opening a gap and extending a gap. When we open a gap, and here we are looking at protein sequences, this is of course very hard, because to open a gap, we need to have a DNA mutation of 3 base pairs. Because we have codons on the DNA, which are translated into protein, and we need 3 nucleotides per codon for 1 amino acid. If we want to remove 1 amino acid, we must remove 3 nucleotides. And what is the possibility to remove not 1, not 2, but 3 nucleotides? It's very low. And therefore, to open a gap is very, how do you say it, penalized. So we have a very bad score for opening a gap. When we extend the gap, we have a lower score. But starting a gap is very difficult. As you can see, these scores are different than just +1 for a match, -1 for a mismatch, and -11 for a gap. We can see that even for matches, for example, matching to valine has a score of 4, but matching to tryptophans has a score of 11. There is a difference there. One match has a higher score than another match. And this is because we use scoring systems. So we have a look in evolution how mutations are actually created, how one amino acid residue is changed into another, and we can use this evolutionary information in our scoring system. So we don't invent scores, but we use information from sequences and databases from multiple organisms where we can actually see the mutations, and we look at how mutations actually can happen in evolution. The first model that we have is the Dayhoff model. And the Dayhoff model resulted in a scoring matrix called the PAM matrix. PAM stands for accepted point mutation. Not point accepted mutation, but accepted point mutation. They used the abbreviation PAM because Appen doesn't sound well. That's the only reason that they used PAM. It should be Appen, maar dat bakt niet. What is a PAM? A PAM is an amino acid substitution that is retained by natural selection, meaning that we have a mutation in the DNA. The mutation in the DNA results in an amino acid change, and this is retained in the organism, meaning that natural selection is not filtering it out. It is something that is allowed, maybe is good, and we can find it in the population. This is a PAM. For the Dayhoff model, we can use these PAMs. If we see a PAM, we know this is a change that is allowed in evolution. We can use this information in our scoring matrix. And for the Dayhoff model, they had to look at a little bit more than 1,500 changes in a lot of sequences and families. They used all this information to create a scoring system, and therefore they created, using 1% change, a PAM1 matrix. So PAM you can also use as an amount of evolution. If we have one PAM of evolution, we have an evolutionary interval in which we have one mutation in 100 residues. Of course, I'm talking about protein sequences here. For the PAM matrix, we get this kind of scoring matrix, which looks like this. We can see that some mutations are not observed. It's not observed. We can see that other mutations are well observed. We can see that a lot. And by using this information, the number of PAMs that we have, we can create scores. If we see that a mutation happens a lot, we can score this high. This is actually happening during evolution. Now, of course, PAM matrices are actually for sequences with 99% identical residues. So these were created from sequences with 99% identical residues. We can use them for sequences starting from, I think it's somewhere in the 80% identical residues, and the PAM matrix, PAM1, is very good. But if we have sequences with a lower percent identity, which are more diverged, a PAM1 matrix is not good to use. Because this matrix was created using sequences with a high percent identity. Then we should have a PAM matrix with a higher PAM number, for which we don't have one allowed substitution in 100 residues, but maybe more. Maybe we can go to, for example, 250 allowed substitutions in 100 residues. Is that possible? Can you have 250 substitutions in 100 residues? I give you a protein sequence with 100 amino acid residues, and I ask you to create 250 amino acid substitutions. Is that possible? Of course. So you can mutate the same residue multiple times, or you can even mutate it into another residue and then mutate it back. These are two mutations, and it looks like no mutations. So we can actually perform 250 mutations, and we will still have 20 to 30% identical residues if we have 250 amino acid substitutions between two sequences. This will still have some recognizable similarity. How do they create this PAM 250 matrix? They were a little bit lazy, I have to say. They used math, and they multiplied the PAM 1 matrix 250 times with itself. You can do this. And it worked. And it's useful. So it's not something that they tried and did not work, so it actually works. So you can multiply the PAM matrix with itself, and in this way you get an idea about how we can have different scoring matrices for different types of identity. Another type of scoring matrix that we have are the Blossom matrices. Blossom matrices, if you ever have used BLAST or another sequence similarity tool, you will see that Blossom matrices are the default matrices that are used. But they were created later, after the PAM matrices from Dehov. The Blossom matrices originate from the blocks database. So there is a database called the blocks database, which contains protein families, and they had a very similar setup as with the PAM matrix. So they look at ungapped aligned regions of distantly related proteins. They found, for example, a protein family with 62% identical residues. And then they used the information about substitutions in that family to create the Blossom 62 matrix. Here, every Blossom number was created from a different input data set. So they did not use matrix multiplication as they did with PAM matrices. So every Blossom matrix is from a different input data set, different sequences. If they want to create a Blossom 50 matrix, they use sequences with 50% identical residues, have a look at the substitutions that we have there, and use that information in the scoring matrix. This is how they create this. So therefore Blossom stands for the blocks substitution matrix. So from the blocks database. Here we can use, for example, Blossom 62 for aligning sequences with 62% or more identical residues. If they are less conserved, have less identical residues, we use a lower Blossom number. For example, Blossom 50. And that's why if we compare Blossom numbers with PAM numbers, we can see that we use high PAM numbers for very divergent sequences, but in the same time, or at the same time, we can use a low Blossom number. If we have a very low PAM number, we can use a very high Blossom number, so they correspond. Of course, low PAM numbers mean less divergent, high PAM numbers mean more divergent. So more divergence between two sequences, less percent identical residues. Most of the time we make use of Blossom matrices. We always try the default matrix, and if that doesn't work, we can try another matrix for more diverse sequences. So most of the times we start with a Blossom 62 matrix, and if that doesn't work, we can try, so we start with Blossom 62, and if that doesn't work, we can, for example, try a PAM 250, which is for more diverse sequences. And maybe then we can still find a hit, because this matrix was created for divergent sequences. And this one was created for more conserved sequences. So it is important that you try to match the scoring matrix with the type or the amount of divergence that we have, but by default we use a Blossom 62 matrix, by default. If we have a scoring matrix to actually score the pairwise sequence alignment, we can use that scoring matrix in the pairwise sequence alignment itself. Now, in general, we have two large groups of pairwise sequence alignments method. We have local alignment methods and global alignment methods. The global alignment methods mean that we have two sequences which we want to align, and we want to align the full-length sequences, meaning that we want to start at the beginning of every sequence and at the end we want to finish. Every residue needs to be aligned with something. This can be a gap, this can be another residue, but every residue needs to be aligned. Of course, if we look at sequences, most of the times we have conserved domains, but we also have more divergent variable regions, which are less conserved. And this can be a problem for pairwise sequence alignment, that it's not possible to actually align this in a good way. Therefore, we can use local alignment. Local alignment means that we will only focus on the regions that are most conserved in my sequences. If we have, for example, at the 5 accent or at the 3 accent of my sequences, we have some more variable regions, we can just remove them because they will not align very well. We only align the regions that are more conserved. We will use pairwise sequence alignment in the next chapter for BLAST, basic local alignment search tool, and there you already know, due to its name, that this will make use of local alignment. The default algorithm that they created results in an optimal alignment, meaning you will get as a result the best possible alignment between two sequences. And for global alignment, this is called the Needleman-Wunsch algorithm. Sorry, there are some names. And for local alignment, this is called the Smith-Waterman algorithm. So these are the two first alignment algorithms that allow you to get the best possible alignment between two sequences. Which means you can try other combinations of aligning two sequences, but you will not get a better result. Based, of course, on the scoring system that you use. New methods, for example BLAST, also makes use of local alignment, but cannot make use of the Smith-Waterman algorithm, because this will be too slow. And therefore they need to make a heuristic algorithm to make that algorithm faster, with enough accuracy. So they need to make some changes to the algorithm to make a faster algorithm which we can actually use to search a database. It's a lot of information for today. No? In global alignment and in local alignment, I propose that you have a look at the course slides and at the syllabus on how this algorithm works. This is always with the same steps. So we start with creating a matrix in which we actually can place two sequences against each other. Then we have a 2D matrix. Then we use our scoring system to score the matrix. And then we trace back the alignment in our scoring matrix. And it's because we fill in the whole 2D matrix that we have the optimal alignment. We can actually find the best alignment because we consider all possibilities of aligning these two sequences. This is the same for global alignment and the same for local alignment. The only thing that is different is that here, the scoring is a little bit different. If we, for example, introduce a gap, we get a negative score. And in global alignment, we can get negative, 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 and we have negative values. The only thing that is different here is that you can never get a negative score. Meaning, if we have a gap, and you add, for example, the score -2, we will reduce the -2 to a 0. And this way there, our alignment stops. You will never go back again. This is a little bit different than we have here. So with local alignment, we never have negative scores in our scoring matrix. With global alignment, we have negative scores. The traceback is also different. The traceback for a local alignment starts with the highest score until you go back to 0. With global alignment, you need to align everything, so there we start at the end and we try to go back to the beginning. So we need to cross or actually go through the whole matrix. And this is not the case for local alignment. There it is sufficient if we only have one part of the matrix that we look at. So here you can see this is an example of local alignment. Normally, if we go vertically or horizontally, this means that we introduce a gap in one of the both sequences. If we go horizontally, this means from this field to that field, this means that we introduce a gap in this sequence. Because we align two residues in sequence 1 with one residue in sequence 2. If we go vertically, this means that we have multiple residues in sequence 2 that align with one residue in sequence 1. This means a gap in sequence 1. You should think if you go in this direction, this points towards the top, and there you will have a gap. If you go in this direction, this points toward this sequence, and there you will have the gap. So to know where the gap is, you can actually indicate the direction that we have here. Normally, for global alignment, if we go horizontally or vertically in our scoring matrix, this means that we will get a negative score. So we go from 0 to, for example, -1, -2, -3, -4. For local alignment, we reduce this to 0. Applications are, for example, BLAST. So it's based on the Smith-Waterman, but it is not the Smith-Waterman algorithm because we need to make it faster. So we need to make some changes, but we will see this later to create a faster algorithm. If we look at the result, and this is the last theoretical slide before we make some exercises. If we look at the result, we will get the alignment of our sequences, but we also get an e-value. And the e-value is similar to the probability value p. I don't know how good your statistics knowledge is. Do you know what a p-value is? It's a chance value of incorrectly.
The chance that it's correct and it's not correct. The chance when it's.
So normally we work with hypothesis. Hypothesis, the null hypothesis, indicating in our situation that two sequences are related. This means they are homologous. The alternative hypothesis states that two sequences are not homologous. The p-value, and here this is called the e-value, gives you the probability of incorrectly rejecting the null hypothesis. Which means if we have a very low e-value, that we are sure that they are homologous sequences. This is for p-value. For an e-value, this is a little bit different. For an e-value, they actually have a look at what is the score of your alignment. We have an alignment score, and they ask you how big is the chance that we can actually get this alignment score when we have, or when we don't have homologous sequences. This is what we test here. So how big is the possibility that we get this alignment score when these two sequences are not homologous? And this is given with the e-value. Also here we have a cutoff value being 5%. If our e-value is lower than 0.05, we can say that we have enough confidence that these two are actually homologous sequences. For example, for beta-globin, myoglobin, only 26% identical residues. We have an e-value of 10 to the -3. This means that we have a possibility of 1 in 1000 that we can have this score by chance from non-homologous sequences by randomly placing two sequences together. So this is a possibility. So we use the expect value as a probability value. The lower the e-value, the better we are confident that we have an actual homologous sequence alignment and not by chance. Let's make some exercises. There are only a couple of exercises which I want to show you, and we still have some time available. Let's start with the first exercise, and I made it easy for you. Normally I will not make it easy for you, but here you will get the accession numbers. So you get the website identifiers, NP and so on, NP and so on. We know that they are homologous sequences. I already showed you, but they have a very low percent identity, meaning using their 3D structure we can recognize that they are homologous sequences, but on sequence level this is hard to detect. Here I have myoglobin and beta-globin. What is the relationship between these sequences? How do we call this? Human myoglobin and human beta-globin. They are homologous. It's in the first line. But what are they? Are they orthologous or paralogous sequences? Paralogous? Yes. They are in the same organism and thus originate from a gene duplication event. So both are paralogous sequences. Now we can try to use the BLAST pairwise sequence alignment utility to align these two sequences. Where can you find this? Google BLAST. So I can search Google for BLAST, and the first hit that I get is the basic local alignment search tool. This also means that if I will align these two sequences, this will be a local alignment, not a global alignment. So I will have a look at the most similar region of these two sequences, and I will only show that most similar region. As you can see, there are different possibilities. We can align nucleotide sequences, we can align protein sequences. Normally we use this to search with a sequence in a sequence database. Today we're going to just align two sequences. As I have two NP numbers, I know that these sequences are protein sequences. Therefore I use the protein BLAST. To actually align these sequences, I use the align two or more sequences functionality. So we have here an input box, but we also have a checkbox that allows you to align two or more sequences. If I check that box, I can have two input fields. My first input field is called the query. I try to match this sequence on top of this sequence, which is called the subject. I can paste my FASTA file here, so my FASTA sequence, so with the bigger than sign, the identifier and the sequence itself, or I can use the accession numbers. So as I have the accession numbers, I can use them. I will start with human myoglobin, NP underscore 005359. I can enter my second accession number in the second input box. This will be beta globin, NP underscore 000509. I use default settings. There's only one thing that I will change, and this is this box. It's a second checkbox, allowing you to show the results in a new window. Because if something of your settings is wrong, we can still change the settings without having to fill in the form again. So show results in a new window, and then we select BLAST. As this is only a comparison of two sequences, this goes very fast, but you can see, although we know that they are paralogous sequences, we cannot find significant similarity. And this is a problem. Why is that? Because they are very divergent sequences, and thus the scoring matrix that I use may not be good for this type of divergence. Where can I find the scoring matrix? I can select the algorithm parameters. And if I look at the algorithm parameters, I see that I use a BLOSSOM62 scoring matrix. This is of course for protein sequences. You will not find the BLOSSOM or the PAM matrices for nucleotide sequences. With this BLOSSOM62 scoring matrix, I expect that my sequences have 62% or more identical residues. And I know that this will not be the case for these two sequences. So I can change, for example, my 62 matrix to a PAM250 matrix, which says that my sequences will have 250 substitutions in 100 residues, which is a lot. I change my matrix, and then I BLAST it again. And here you can see that we actually have similarity. This is my query. And you can see that this region starting here and ending there actually aligns with my subject, with my second sequence. You can see the score with the value. We want red score, but we have a blue score, which is not that good. And we can see that we have an actual coverage of my query of 88%. And of this 88%, we have 26% identical residues. the E value equals 2 times 10 to the minus 13. So we are very confident that we cannot find this alignment by chance. That this is actual homologous sequences which align. You can also see that we have 26% identities, but 51% positives. We call this similarity, which means that we can also take into account the biochemical properties of my amino acid. If you mutate a hydrophobic amino acid into another hydrophobic amino acid, we can say that this is a match, but not a perfect match. So it's not an identical, but a positive match. So if they have the same properties, we can take that into account. And here you can see our actual alignment, indicating that they start at position 11 in my query and end at position 146. In my subject, they start at position 12 and ends at position 146. Does this work? Ja. 2 keer 10 tot de min 13. Yeah.
I missed the part about the positives.
So the positives is when taking into account the biochemical properties of the amino acids. If they belong to the same group, for example, hydrophobic or positively charged or negatively charged amino acids, then this is a positive match, being they are similar amino acids, but they are not identical. But of course, for our scoring matrix, this is useful. Because it is likely that if we have a mutation of a hydrophobic residue, that it will be mutated into another hydrophobic residue, and not into a hydrophilic one. Let's have a look at the second one. For the second exercise, we will compare human mitochondrial DNA from modern humans to the extinct Neanderthal DNA. First, we will obtain the nucleotide sequence of a mitochondrially encoded gene called cytochrome oxidase, and next we perform a pairwise sequence alignment. I propose to start with Neanderthal because I know that there is less information available for Neanderthal. Where can I find the Neanderthal data? How can I find Neanderthal? Where do I start? So this is for a specific organism. Taxonomy browser. So this is the easiest way to do this. So I search for the taxonomy browser. In the taxonomy browser, I can search for Neanderthal. I am redirected to the Homo sapiens neanderthalensis webpage, and there I am looking for the nucleotide sequence of the mitochondrially encoded cytochrome oxidase. Here I can actually redirect to gene to not have redundant information. So I select the 37 that I can find next to the gene. And as you can see, the filter for Neanderthal is automatically applied at the NCBI gene webpage. Here I am looking for cytochrome oxidase. And as you can see, cytochrome C oxidase subunit 2, cytochrome C oxidase subunit 1, subunit 3, cytochrome B. There are only a couple of matches available, and then some tRNAs. So cytochrome oxidase, you can choose subunit 1, 2, and 3. Maybe we can start with 1. You can choose this. So I'll choose COX-1, so the cytochrome C oxidase subunit 1. Let me now first have a look if I can find this gene also in humans. So cytochrome C oxidase subunit 1. Therefore, I go back to the taxonomy browser. So I open this in a new tab. I go back to the taxonomy browser, and here I search for human. I select Homo sapiens. now I want to go to gene, but here I select direct links to not include the Neanderthal, which is a subdivision of Homo sapiens. So I select the last column for gene. Here I can search for, and let's have a look, this is called COX1, so I'll try COX1. So you can use Boolean operators to match multiple fields. So and the organism and the name of my gene. I'll search, and I can find mitochondrially encoded cytochrome C oxidase 1, which seems to be relevant, I think. An alias, therefore, is also COX1. So I'll go to that record, and here I'm looking for the nucleotide sequence. Where is my table of content? It's available here. I can go to NCBI reference sequences. I can do this as well for Neanderthal, NCBI reference sequences. And you can see that for Neanderthal we only have genomic DNA available and the protein sequence, but we do not have messenger RNA sequences available, which is of course logic for an extinct animal. So here we will need to make use of this genomic sequence. We can find this in FASTA format, or we can use the accession number. Also for the human, it's better to use the accession number or the FASTA sequence of the genomic DNA. So let's go to the FASTA sequence. I'll also do this here. I'll go to the FASTA sequence, and then I can start my BLAST pairwise sequence alignment utility. So I go to BLAST. If you have used it before, you can directly go to BLAST. And now we are comparing nucleotide sequences. So we select NCBI nucleotide BLAST, sorry, not NCBI nucleotide. So nucleotide BLAST. Again, here we select line 2 or more sequences. And now we can copy the nucleotide sequences in FASTA format from both organisms. So I can, for example, start with my human. Copy, paste, and I can do the same for Neanderthal. Copy, paste. You can see that this does not use the default, or it's actually not default because this is the default one. The default BLAST algorithm for nucleotide sequences is for highly similar sequences. This is called MegaBLAST. Normally we should use the BLAST-N, but we can try this with MegaBLAST. Show results in a new window. If I look at the other parameters, you can see that we cannot select a BLOSSOM matrix or a PAM matrix here, because these are created for protein sequences, not for nucleotide sequences. We can select BLAST. And let's have a look. This is actually quite good. So you can see that the input of my query was 1542 base pairs. This is the same for my subject. So my human and my Neanderthal have the same length. And if I look, this is 100% coverage with 99% identical residues between human and Neanderthal cytochrome C oxidase at the nucleotide level, which is actually quite good. Of course, this is in evolution something that happened recently in Neanderthal. It's not like the speciation between humans and another organism, so this is very recent, and therefore we have a lot of sequence similarity, a lot of identical residues. The expect value equals 0. meaning that you have no chance that this is due to randomly placing two sequences together. We will not do the last exercise, it's an easy exercise. Hippopotamus, pig, whale, so 3 organisms. What you need to do is just look them up in the taxonomy browser. You can search with their trivial names, so you can search the taxonomy browser with hippopotamus, and then you will find the Latin name for that organism there. There you can try to find a protein sequence of hemoglobin for each of these 3 organisms. So you go from the taxonomy browser to NCBI gene for that organism, and there you search for hemoglobin. You first do this for the 3 organisms, and you try to find the same hemoglobin in all 3 organisms, and then you can compare them. It's pairwise comparison, so you need to make a couple of comparisons. So in total you will have 3, it's already late, 3 pairwise comparisons. And using these comparisons we can try to distinguish the relationship of these organisms. So here I restricted your search to protein sequence of hemoglobin. Know that I can leave out the hemoglobin, and then you need to decide for yourself which gene or which protein will I use. If it is to detect homology, we use protein sequences. If it is to detect a relationship, we will see this later, it's easier to use nucleotide sequences because more information is available in these nucleotide sequences, which is of course filtered out in these protein sequences. Okay, that was it for today. Wish you good luck, and good luck next week with the exam for Lab Technologies. Here protein. Yeah. So we search for NP numbers.